Hello, welcome to Chapter 2 of Bio23 Infectious Disease. In this chapter, we're going to talk about modern medicine, which has evolved as a result of post-germ theory of disease. Basically, that diseases are caused by microbes and not by bad air, bad karma, or an angry God. All of this started to change with scientific evidence that was discovered and published at the end of the 1800s and the beginning of the 1900s. There were two scientists in particular that had a strong impact in this field. They were Louis Pasteur, a Frenchman, and Robert Koch, a German man. Because they didn't speak the same language or read each other's publications, they weren't fully aware of what each other was working on, but in fact, they were both working on very similar things. And that was this field of germ theory of disease, that microorganisms were the cause of infectious disease. Let's begin by discussing Louis Pasteur's contribution to this field. One of his major contributions was in the field of spontaneous generation. He conducted a very famous experiment that he repeated many times using many different conditions. This experiment entailed using what was referred to as a swan-necked flask. This flask contained liquid that had hay or beef or other materials that would provide nutrients to microorganisms. He then boiled this liquid for an extended period of time in an effort to kill everything that was contained within it. He let the flask cool and allowed time to pass, and during that time, that liquid continued to remain clear, indicating that no living system was able to grow inside. He used the same flask, and at any point in time, if he ever broke off the long swan neck to it, found that within days, that liquid substance would turn cloudy, indicating microbes were growing. This was a controlled scientific experiment that he ran many times. And at the end of all of this, his conclusion was that the long swan neck, while it allowed for air to pass in and out, did not allow tiny little microbes that were known to exist to travel up into that long neck. And that's because even microbes, as small as they are, respond to gravity. So they would enter the swan neck and settle at the bottom and not be able to travel up the long distance and into the liquid solution. But as soon as that neck was broken, the microbes could enter and grow. He's also known for generating a similar technique in liquid that people would consume. This started with beer and wine, and eventually segued into using it for milk and juice and cheeses as well. This process is now referred to as pasteurization, and what it entails is rapidly heating a liquid to warm or hot temperatures and then cooling it down after a short period of time. Pasteurization does not have as its goal to sterilize the liquid, as we saw in the previous experiments. The goal of pasteurization is to reduce the number of microbes in the liquid. That is to allow for a longer shelf life of these various liquids, while it does not change the flavor of the liquid, the beer, wine, or milk, very much at all. Arguably, this process of pasteurization, especially in relation to juices and milk have saved millions of lives and is still an important part of a healthy lifestyle. Louis Pasteur, knowing about these microorganisms that lived on the surface and inside of many solutions and substances, then hypothesized that perhaps the reason why humans and animals get sick is because of the presence of these microorganisms. As a result, he developed what we now refer to today as the germ theory of disease. In using the germ theory of disease, he connected rabies, a symptom, to a microorganism that we now know of as a virus. In so doing, he was able to also generate a vaccine against this disease that is still used today when and if anyone gets bit by a rabid animal. An individual in Germany had an even greater contribution to supporting the field of the germ theory of disease. His name was Robert Koch. Robert Koch's seminal work in the germ theory of disease field was to generate Koch's postulates. Koch's postulates are essentially 4 steps of an experimental process that, if every single step is held true, can connect a microorganism to a specific disease. Koch's postulates are listed here, and I would like you to know them. The first step is to identify all of the individuals with a specific disease and find a microbe commonly found in each. If the same microorganism is found in every case of the disease, that microorganism is then taken to step 2 and isolated in a laboratory. A pure culture is generated in which the only microorganism growing in that culture is the suspect microbe. Step 3 is to then take this purified microorganism and inject it into a laboratory animal. The laboratory animal is observed for signs of that specific disease. Finally, after the animal shows symptoms of the disease, the 4th step is to re-isolate that specific microbe in the lab once again. Koch's postulates have been used extensively in demonstrating the connection between living organisms, microorganisms, and specific infectious diseases. And it still continues to be used today anytime a new or emerging infection arises. However, there are some issues with Koch's postulates. One of these issues with Koch's postulates is that some microorganisms grow extremely slowly or not at all in the lab. If you cannot grow a microorganism efficiently in a lab, then postulate 2 and postulate 4, wherein we grow and isolate the microbe in a lab, cannot be fulfilled, and Koch's postulates cannot be used. Your book references a good example of this, Mycobacterium leprae, the now known causal agent of leprosy. But syphilis is another example of these that cannot be grown in pure culture in the lab. Viruses could also arguably fall in this category, since viruses cannot be grown by themselves on a petri dish. However, if a host cell line can be grown in a purified form in the lab, then viruses can grow inside of them. So we can still use Koch's postulates with viruses as long as we have the host cell line. Secondly, another issue with Koch's postulates is animal models. Some animals simply do not get sick with the same symptoms, or sometimes at all, from diseases or microbes that infect us. A perfect example of this is HIV. HIV, which stands for human immunodeficiency virus, only infects human beings. Generally speaking, it has to be altered in order for it to effectively infect our closest relative, the chimpanzee. Without a good animal model organism, Koch's postulates, specifically 3 and 4, cannot be fulfilled. The germ theory of disease was gaining mounds of support from Louis Pasteur, Robert Koch, and many other scientists at the time. So much so that health agencies began implicating microbes in the causal agent of disease, and public health departments, specifically in the United States and Europe, were gearing up toward defending humans from these tiny invaders. In a very famous case in 1906, a specific individual was singled out by the Public Health Department of the United States as being the cause of spreading an infectious disease. This is the story of Typhoid Mary. Typhoid Mary was a cook in New York and the East Coast. Her full name was Mary Malone, and she was an Irish woman, an immigrant who was unmarried and could only make a living by cooking for families in New York. Over several years, many of the families for whom she worked ended up getting sick with typhoid fever. Typhoid fever is caused by a bacterial infection, Salmonella typhi, transmitted by what's referred to as the oral fecal route. When someone poops, gets some fecal material on their hands, does not wash their hands, and then transmit that disease to someone else who eats it. In cooked food, this wouldn't be a problem, but Mary Malone's specialty was peaches and cream, not cooked, thereby allowing Salmonella typhi to be transmitted so easily. She was implicated in the illness of roughly 50 cases, 2 of whom had died, when the Public Health Department finally identified her as the transmitter of the disease. She was forcibly asked to stop her employment as a cook. But Mary Malone, not believing she was the cause of the disease, changed her name, and a few years later began working in a hospital. There, she ended up infecting a couple dozen more individuals, including resulting in 2 deaths. The reason why she didn't believe she was the cause was because she was otherwise healthy. However, samples taken from her feces and eventual samples taken from her appendix and spleen indicated that she was carrying copious amounts of the Salmonella typhi bacterial strain. She ended up being quarantined and essentially imprisoned on an island off the coast of New York. There, she lived out the rest of her days in a hospital field, unemployed and dejected. We'll continue on this discussion about the germ theory of disease and modern medicine in the next section.